students, let us discuss about the today's class, especially the cell biology. For your syllabus, paper 1, you are supposed to face the unit 1. That unit 1 consisting of the cell. There are two types of cells. One is the prokaryotic cell and second one is the eukaryotic cell. In today's class, in brief, I will explain what exactly the prokaryotic cell is. Here, while considering the prokaryotic cell, take for example, this is the structure of bacteria, and this structure of bacteria is mainly consisting of three main parts. One is the cell wall, second one is the capsule, and the third one is said to be the internal core of the bacteria. So the outermost covering layer of a bacteria is called as the cell wall, and this cell wall is very hard and rigid, and this cell wall is mainly consisting of the peptidoglycan. In case of the gram-positive bacteria, 90% of the cell wall is mainly made up of the peptidoglycan, whereas in case of gram-negative bacteria, that cell wall is mainly made up of only 10% of the peptidoglycan. So this cell wall will have very strong and rigid. That rigidity of the bacteria, it is due to the cell wall here. That cell wall is followed by the capsule. Capsule is always gives protection to the bacteria. So immediately after the cell wall, there is one more layer called as the capsule. That capsule, the main function of capsule is the production. And this capsule is followed by the one more membrane called as the plasma membrane. And what you are supposed to see here, this is said to be the plasma membrane. This plasma membrane, and here and there you can see the inward folding of the plasma membrane is called as the mesosomes. And these mesosomes are the site of the respiratory enzymes do present in case of mesosomes. Hence, most of the catabolic and anabolic reactions are taking place in this area only, or in the mesosome only. And here and there you can see the hair-like structures or outgrowths. They are called as the pili. They are also called as fibriae. These pili are also called as the sex pili because they are mainly involving during the transformation of genetic material from one bacteria to another bacteria during the conjugation here. And here you can see the pair of flagellum. These flagellums are mainly meant for the locomotory organ here in case of bacteria. And inside the plasma membrane, you can see the cytoplasm here. That cytoplasm is very rich in carbohydrate, proteins, lipids, and organic and inorganic salts, including vitamins. And here and there you can see the polyribosomes here. And these are all small units are called as the ribosomes. That ribosomes are always 70 years with respect to the prokaryotic organisms like bacteria. So these ribosomes do involve during the protein synthesis, especially in the form of the polyribosomic condition. And it is also consisting of the plasmid. This plasmid is a extra chromosomal double-stranded DNA present in the bacteria, and these do carry the R factor and F factor in case of the cloning process here. And meanwhile you can see the nucleoid. It is without nuclear membrane. There is a genetic material called as the nucleoid. So this nucleoid is responsible for transformation of character from one bacteria to another bacteria through the conjugation here. So this nucleoid is totally the devoid, or I can say the lack of the nuclear membrane is present, and it is always embedded in the cytoplasm of the cell. So this includes the prokaryotic cells, especially the bacteria. Hope you can understand well with respect to bacteria. If you find any difficulty, read well. I have already sent the notes in WhatsApp group. Read well. If you find any difficulty, you please ask me in the discussion section. Thank you.